translation today involves federal government. So our first speaker is Carlos Peña, who's going to talk about requirements for devices by the FDA. And since it's fairly small and I think more people will come in, we can make it somewhat informal. And if you have any questions, just speak up. And this is what we did yesterday as well. So Carlos, you want to start? Or? Yeah, I don't think those are his slides.
Carlos Peña, Director for the Review Division of Neurological and Physical Medicine Devices at the Center for Devices at the Food and Drug Administration. Oh, sorry about that. And I was going to give go over a couple topics about our Center for Devices, including a backgrounder, sort of telling you a little bit about regulatory pathways. Then going into some more detail about the clinical studies that collect data for medical devices. Then sharing with you a little bit more about some activities that we're having, some recent activities at the center that talk about medical devices in general. And then I'll close with some best practices and closing remarks. But if the very first slide, or couple slides now. They should be pulling it up shortly. I don't know. I know dad jokes. You know, what do vegetarian zombies eat? They eat grains. So, you know, brains. So, yeah, that's about as good as it gets. There we go. So, appreciate it. So there's the outline, sort of a background. There are some pathways, points to consider for medical devices, which is going to be parallel to those points that I talked about yesterday with regard to neurostimulation devices. Some activities highlights, and then a few best practices. So our vision at CDRH is that patients in the US have access to high quality, safe and effective medical devices of public health importance first in the world. We take that, excuse me, we take that vision statement very seriously. You know, our goal, I think when we're around a table with sponsors and companies and patient groups and other agencies, other government agencies, we're all trying to, I think, get safe and effective products that are meaningful to patients, clinicians, and their families. So I'll share with you some data in just a little bit. For definitions, a medical device is a lot of things here on the slide. An instrument, apparatus, implement, you know, goes on and on. And so you're probably saying, you know, Carlos, really, what is a medical device? And so if it diagnoses, you know, high level, if it diagnoses, prevents, or treats a disease, it may be a medical device. And this usually occurs in the absence of a primary chemical action. You can also have a medical device even if it's not labeled a medical device. So, you know, it's hard to skirt around sometimes the regulation for medical devices, even if they don't label it as a device. We take a risk-based approach to the oversight. So here is a slide that talks about the different classifications, which I'll get into more details in the subsequent slides, but there's 3 types of medical devices: class 1, class 2, class 3, based upon risk. Class 1 are low risks, generally exempt from our oversight. Class 2 are moderate risks, requires what's known as a 510K pre-market notification, also known as the 510K. You can also have de novos, and I'll talk about both those pathways. Those are for our moderate risk devices. And then the highest risk device classification is the class 3, requires what's known as a PMA, a pre-market approval application. And we provide oversight along these 3 classes with both general and special controls, and in some cases, clinical data is needed to make a marketing decision. So here's a little bit more, we're going to go a little bit deeper now. So class 3, generally PMA, pre-market approval. The center receives several dozens of PMAs each year. Those are our highest risk. Class 2, 510K, pre-market notification. These are for like me devices. So there's something on the market. If someone has a new idea, wants to target a different population with the same technology or has a slightly tweak to the technology, they might submit a 510K application, a pre-market notification application. It's comparable to the ANDA of drug land. We receive several thousand 510Ks each year. Not in the neuro space, but as a center. De novos, those are devices that aren't comparable enough to something on the market. They are low to moderate risk. This generates a new device classification, and once we get that device to market, that will serve as the predicate for new devices, and those new devices will go into the 510K. So the de novo gets us off the ground, and the 510K process can allow for that initial decision to increase the numbers of that area with 510K submissions. We just had a de novo panel meeting last week, the Neuro AD panel meeting, and it was a TMS, transcranial magnetic stim, and cognitive dual treatment approach to Alzheimer's, mild cognitive, and Alzheimer's dementia. It's a very active space. We're seeing an uptick in the neurophysical medicine devices. Last is the humanitarian device exemption application. These are intended for diseases or conditions that affect a very small, relatively rare population. And it's the regulatory bar for HDE is safety and probable benefit. Whereas in the PMA, the highest risk devices, HDEs are class 3 too, but the PMA, there the bar is a reasonable assurance of safety and effectiveness. So here's now a further dive into those 3 pathways, PMA, 510K, and de novo. So PMAs, approved for class 3 devices. The majority of these PMA applications are supported by clinical data under what's known as an investigational device exemption. That's the application that you use to study a device in a clinical study at a site, at an academic site or a hospital. These applications are subject to annual reporting, so they have post-market requirements. They may have a post-approval study. Many times there's a couple hundred patients that are involved in at least the neurophysical medicine device area, and we may have questions about the long-term use, the long-term effectiveness, as well as the safety. So in some cases, we'll work with sponsors to mandate a post-approval study. And there's also manufacturing, monitoring, and inspections of that site. The pre-market notification program, so this is, again, the like me devices, 510K submissions to the agency. The bar here is that you must establish substantial equivalence to a legally marketed predicate device. So we'll take a look at the IFU. Is it the same IFU? Is it comparable technology? So let's say you have a neurostimulator. Is the output comparable to what we already have on the market? If it does not raise any new safety or effectiveness questions, it could potentially go through the 510K notification program, which takes about 90 days to get to market. And those are goals. Now, the third program that I talked about is the Denovo Pathway. So we have the PMA, the 510K, here's the Denovo Pathway. These, again, are low to moderate risk, do not fall within any classification regulation. We haven't seen these types of products before. Previously, what people did was they went through the 510K program, and they got a negative decision, then they had to submit a de novo. We've updated our processes so that sponsors can go directly to a de novo pathway. And we usually write what's known as general and special controls to help ensure the reasonable assurance of safety and effectiveness for those products. And that anytime you make a de novo decision, something gets to market, it generates a new device regulation because you have a regulation for that particular product. Many times during the PMA, the pre-market approval, and the Denovo Pathway, we have an option for expedited access. And this is intended to address an unmet medical need for a life-threatening or irreversible debilitating disease or condition. It's voluntary. It's the expedited access pathway. More recently, it's called the breakthrough pathway. And what happens here is that we work with those sponsors to lay out the full device development program to get that product to market. We'll invest with our limited resources, as much resources as possible, to making sure that we get that through that review for that product, whether it's Denovo or PMA. In some instances, it can also be applied to the 510K pathway, but it's hard to shave off days when there's a 90-day review clock. It's more easier to shave off days when there's the goal is 120 days for de novos and 150 days for PMAs. And the criteria for the EAP, the breakthrough designation is life-threatening or irreversible debilitating disease or conditions. We provided a data development plan, so it really lays out the requirements for companies and sponsors to get their product to the market. This is really good for understanding the costs, understanding the non-clinical and clinical requirements, understanding the business decisions that might be needed for any particular product. So when is clinical data needed? That's the next big question that gets very expensive. So for PMAs, I mentioned they're typically needed. Clinical data is typically needed in those applications. For de novos, typically needed, but not always. And for 510Ks, it's typically not needed. And so you're probably saying, there's a lot of provisos, addendums, there's a lot of, really, Carlos, when is it needed? And so we work with sponsors to identify and clarify what the needs are for their particular device in what's known as a pre-submission. And that's a free process, so we encourage folks to use it as much as they'd like, within reason. But you can get feedback through the pre-submission process on the clinical data needed to support an application to get a device to the marketplace. And we've seen a lot of uptick in those pre-submissions. They usually come in, we take about 2 months to review them, and then we set up a meeting or a telecon or provide written responses to their questions. And the questions can be anything. Is this the right data set for an animal? Is this the right animal data set to move to a human study? Because many times you'll need some animal data to get to a human study. The IDE permits the clinical investigation of devices. So sponsors, companies will come in. Many times academic PIs, academic faculty will come in with an IDE request to permit using deep brain stimulation for a new indication, or spinal cord stimulation for a new type of pain, or a new type of neurothrombectomy device to treat acute ischemic stroke. We get 30 days to review that submission, and it's not a lot of time. We'd like to approve that submission because the faster a study is up and running, the faster the data is collected. Faster data is collected, faster data we can see, and faster we can make a decision to potentially get a product to the patients and their families. One big question is whether it's a significant risk, because if it's significant risk, that requires an IDE. There's significant risk and non-significant risk devices. For NSR devices, they don't require an IDE. They usually work with their IRBs, local IRBs. But for significant risk devices, it hits one of these 4 criteria. It's an implant, typically for 30 days or longer. It's used in supporting or sustaining human life. Substantial importance in those criteria. And the 4th one, which gives us a little bit of flexibility, is otherwise presents a potential for serious risk to health, safety, or welfare of the subject. So if you're using a toothbrush to brush your teeth, probably not a significant risk. If you're using a toothbrush to do brain surgery, we may have some questions. But we consider the device itself, as well as how it would be used, to make a best determination on the risk, whether it's SR, NSR, and whether it requires an IDE. So here's a little bit of a risk determination by classification chart. So now everyone's familiar with PMAs, Denovos, 510Ks, HTEs, all the lingo. And many times the class 3 devices on the right-hand side, the highest risk devices, both PMA and humanitarian device exemption devices, are significant risk. But you can have class 2 devices that are both significant risk, or they can be non-significant risk depending upon their use. In those cases, for non-significant risk devices, we wouldn't see those products. But we are encouraging sponsors more and more to submit a pre-submission to share with us your development plan to getting that product to the marketplace, because we want to make sure that if you're making a claim about a device that is a class 2 or even a class 1 device, if you're making a claim that it is a cure for Parkinson's disease, it might be able to talk with us about the data that you're justifying that claim. So we're very much encouraging the discussions at the front end of device development rather than when the study's all done and you have a data set. There are 3 types of studies where you collect medical device clinical data from which to put into a marketing application. One is an EFS, early feasibility study. It's a very small number of subjects, less than 10. It's early in development. You may even be tweaking the final device design. And we kind of work with sponsors to sort of get those studies up and running, because that's sort of like an early indicator. We also want our studies to be started in the United States. Many times companies may go overseas because there's the perception that it is easier to do a study overseas than in the US. They usually do some maybe feasibility studies overseas and they come to the US for a pivotal study. We'd like to get those studies started here in the US, so long as safety is protected, the patients are protected. But we'd like to have those studies start here. So we're trying to work with sponsors on the EFS type of study, where we are looking very closely, working with sponsors on both the non-clinical and clinical data that's available about that product. Traditional feasibility studies, a little bit larger, 20, 30 subjects, and then pivotal studies where you collect the evidence for the basis of a marketing decision. That's the data that we would look at to make a decision about a product, whether it can go to the US marketplace. And there are 3 decisions for IDE studies. One is an approval decision, full approval. They're good to go, so long as, along with IRB approval, but they've designed the study well, the outcomes make sense, the sample size makes sense, patients are protected, the informed consent looks good. It's a full approval for them to begin. Approved with conditions means that there might be some issues that we still need to work out. Those should be addressed within 45 days. And then there's a disapproval. The trial may not start, and that's primarily for safety issues. We like to get full approval on our IDEs, and that has to happen in 30 days. That's a really tight deadline for us to look at the application, pull together a team, have the team review the application. Sometimes there are hundreds and hundreds of pages, and then make a decision on how to interact with the sponsor to either address the concerns that we may have, or if there's still time remaining, or is it good to go back with some deficiencies? The study's not started, and we work with the sponsor until they're ready to resubmit. This is what that process looks like over time for the past several years, with in regards to standing up fully approved studies. So in FY11, it took about 400 plus days to stand up the IDE studies. That's the median time. FY13, 14, we had some legislative changes that helped to reduce the times, but in 15 through 17, and again in 18, because the numbers are still being crunched, but it looks like the numbers are tracking very closely to 30 days as the median time. We've been able to stand up our studies a lot quicker, so long as safety is protected in these studies. And that takes both, work on our side, the staff, especially in the neurophysical medicine side, to get those studies up and running. But it also helps, for me and the staff that work in my office to come to conferences here and share with you our expectations, because you know, if people understand what we're looking for with regard to safety and effectiveness, trial design, sample size, outcome measures, patient populations, eligibility criteria, informed consent, then we're able to more easily stand up those studies within 30 days. Sometimes we call up the PIs at an academic center and we say, hey, we have a couple issues to go through, and you know, they're like, you know, who is FDA? What does FDA stand for? You know, and so it's very challenging to work with folks that haven't been sort of submit the application in pre-sub to get some early input, have not interacted with FDA. We try to have those interactions as early as possible, even before 90 submissions are submitted. So early collaboration is critical, get feedback early, and we're able to refine the patient population that's most likely to benefit with the least amount of risk. So here is an array of devices that have come through the Office of Neurophysical Medicine, neurothrombectomy devices, ablation therapies, cognitive assessment software, prosthetics for amputees, devices for migraine, and even the microcatheters. The two end panels have been, those went through 510(k) submissions because there's prior products on the market. There is prior knowledge about those devices. The prosthetic arm was a Denovo submission, as well as the medical device, the migraine device. The ablation therapy, I believe, was a pre-market approval submission, and the cognitive functions following concussion, that was a Denovo submission. Purpose is not to talk to you about the particular data set that was applied, but it's to share with you that each of these reviews involve tailoring the review to the particular risks and benefits and safety and effectiveness of that product. And so you see that's never really, we have mechanisms to get these products to market. The questions that come up are the clinical issues, the scientific issues. You know, how well are patients after using that clot retriever? What is their disability like? Is it better? Is it enough for us to market that product beyond you know, standard of care? That's not a regulatory issue. That's a clinical issue. That's a clinical outcome issue. And ideally, we would have had those discussions in a pre-submission. So there's a couple points I'd like to go through in the next couple slides with regard to medical device testing research. There's typically two phases of development. There's exploratory, which is the feasibility portion I talked about. And then there's the validation, the pivotal study that usually comes with that validation. Sometimes we get just the validation. The data is mixed. It's kind of fuzzy. It's not, you know, may not have met its primary endpoint. We try to avoid those unusual outcomes by having the feasibility study first, which gives us some indication of how to design the validation study. So we do like a two-step here. There are cases where it makes perfect sense to move to a pivotal study with what's known in the literature or overseas, or even some preliminary investigation somewhere, but most cases it involves a two-step process. In the medical device land, it's very different than the drug land. So in medical devices, we are required by law to look at what's called valid scientific evidence, which can range from studies that are open label, single arm, no control, all the way to randomized blinded sham control studies. So that's very different than the typical drug world where they have typically two well-controlled randomized studies. But you know, the devices, they have different needs. They have different study designs. So you know, it makes sense that while the tools may be different, I think it's reassuring to know that the bar for safety and effectiveness is equivalent. It's, and we are trying to make sure that that principle is maintained. With regard to safety, when you do a medical device study, we're interested in all the adverse events being reported. That should be part of the clinical protocol. There should be a safety plan a primary safety endpoint that you should hit. We want to know about the short-term and long-term safety of medical devices, especially neurological devices. And we want to know what happens with the device in the short term. Are there a number of revisions? Are there replacements? Are there explants? Because if there's a lot of that, if there's a lot of revisions, there's a lot of replacements, the risks of the product are beginning to outweigh the benefit. That may dial into our decision process about whether to market that product, give marketing authorization for that product. Same for effectiveness. We look very closely during the design of the study on the endpoints. Are they meaningful to both clinicians and patients? I think in the panel last week, with regard to de novo application for Alzheimer's, there was a lot of question whether the panel raised a lot of questions about the meaningfulness of the changes when you're using that product compared to a sham group. Are the outcomes validated? We can look at non-validated measures, but typically we like to have measures that we know what the variability is, measures that we know what the inter-rater reliability is, measures that we know what change is meaningful with that measure. And there's a number of other things that we'll think about through with sponsors, investigators, about the effectiveness endpoints they choose for both the feasibility, which should have some effectiveness endpoints and preliminary endpoints, and as well as safety, and those endpoints for the pivotal study. Hopefully you agree to those endpoints before the pivotal study started. Because if you start to sort of not reach your endpoint and then start to sort of mine the data, that's going back to an exploratory phase of development. It's not a validation of what you've hypothesized. And so we've gone through some of these issues. Medical devices can be difficult to blind and randomize. I showed you an image of the prosthetic arm. How do you blind that in a clinical study? And it's a little challenging. So, you know, the trials may be different for prosthetic arms that we're looking at, the DECA arm. Device studies are done to support a reasonable assurance of safety and effectiveness. When the study is done, even though you've gone after a particular patient population with an IFU, can you generalize those results to have a broader impact upon the patient community? These are questions that I think would be important to address as the study's done and when the results come in. And then we have post-market activities. I talked a little bit about this. I'm not going to talk too much about this, but there may be post-approval requirements. There are compliance activities, oversights that we are involved in, and surveillance. You know, sometimes we run a study, a pivotal study. It looks good. Safety and effectiveness has been met. But then there are these safety signals that we see when the broader population is using these medical devices. And we may need to refine the IFU post-market. So we're looking at ways to not only make a decision on the pre-market side, usually with limited data sets, but we're also looking at the post-market side as an additional tool that we have to make refinements and adjustments to medical devices that might be in the marketplace to better increase the benefits to patients and reduce the risks. Finally, a couple organizational slides that I'd like to share with you. We're one of the newest divisions in the Center for Devices, relatively new. Like, I think it's been about 5 years, but like at FDA, 5 years is like, you know, really quick. We are one of several divisions, and we're within the Office of Device Evaluation, all that yellow box all the way to the end. There's also a number of other offices in the Center for Devices currently, and we're going through a reorganization now to combine a couple offices, including the Office of Device Evaluation, the Office of Compliance, so that's our post-market office that takes looks at enforcement and labeling and other post-market activities, and combining the first yellow box on the left, surveillance and biometrics, into one office, into the Office of Neuro and Physical Medicine Devices. We hope to go live May 1st. That will give sponsors an opportunity to not only talk with the team on the pre-market side, but they'll be talking to the same members of the team on the post-market side. And so they really get a very broad perspective about how their product's going to be managed, pre-market and post-market, which helps them make decisions, helps their funders make decisions, and it starts to build in institutional knowledge within the team, which I think is a good change that we're making at the center. Currently, we have 5 branches within the Neuro and Physical Medicine Devices division, which is an office. We are breaking out the panel on your left, diagnostics and neurosurgical, into 2 teams. Diagnostics has increased dramatically, purporting to diagnose Parkinson's or epilepsy or brain tumor. That is a big area that's growing, especially with the mobile medical application activities. And then we are also breaking out the physical medicine and rehab into acute and rehabilitative devices. Mentioned pre-submissions, just do one more plug about this. Actually, there's one last plug at the last slide, but pre-submissions, feedback from FDA, you know, there's the story several years ago was like, I don't know who to contact at FDA, I don't know what FDA does. This is an opportunity that I think helps to introduce the agency to folks. And the pre-submission can even be an informational meeting to get the agency aware about the technology that you're developing. We're also trying to increase our transparency about how we look at product areas. So we have pre-submission guidance. The guidance documents are basically ways that we communicate to sponsors and FDA staff sometimes what our thinking is. So there's a lot of guidance listed here. One guidance that came out in 2015 was clinical considerations for neurological devices targeting disease progression. It's a big deal, right? There's some speculation about some of the neuro devices purporting to change the progression, slow the progression of various diseases, and we're trying to keep track with that innovation area. We also just released last month a guidance on brain-computer interfaces for patients with paralysis or amputation. That is also intended not only to spell out what we're most interested in during those IDE studies, but it's also to keep pace with the emerging tech area and foster and support the innovation that we see around, around the country, around the U.S. Mobile medical applications, we have a guidance out. It's been out for a couple of years now, but it's been getting its fair use along the community. These are for applications that we're seeing, at least in the neurospace, to diagnose a condition or a disorder. If it purports to make a standalone diagnosis, we may have some questions. If it's just sort of tracking, becoming a log of symptoms, we may have less questions. But in that guidance, if you're developing a mobile medical app for the healthcare area, we have a number of appendices that provide examples of what we consider to require some oversight, what is under enforcement discretion, and what is not a medical device. So we are trying to take a risk-based approach with the limited resources that we have and make sure that for those mobile medical apps that may be diagnosing a condition, that seems pretty significant to both the patient, the family, and the physician. So there may be some questions that we have about that product versus a product that is just storing information about symptoms. There may be less questions, might even not require some oversight, but I would encourage folks to take a look at that MMA guidance. The second initiative is we're having more and more focus on patients, patient use, patient preferences. We're partnering with different patient groups, looking at different patient-reported outcomes. One of the outcomes of the meeting last week with the Alzheimer's was that the panel was very much interested in more outcomes that were meaningful to patients with Alzheimer's. They thought that it'd be great to, have patients remember their parents or remember loved ones versus remembering a word on some test. So there is more and more interest on behalf of the agency to having patient input, especially on the outcome measures that would be most meaningful to patients. And so there's a couple of guidance documents on this area as well, and some activities that you'll probably be hearing a little bit later today from a colleague of mine, Dr. Tarver. Last slide is last slide is real-world data. We're looking at how we can use evidence from hospitals through case report forms, electronic health records, how that data can be supportive to regulatory decision-making. Many times we get a device on the market, it gets a lot of use. Sometimes it gets some use that's off-label. We'd like to take a look at that off-label use to see if there's any information that we can apply to an IDE study, apply to a marketing decision, or make a decision upon to expand the indications for use for that product. Because there's a lot of information when you look at the hundreds of thousands of records that are in the community or that may be available with our servicemen and women in the military. And so real-world data, real-world evidence is something of interest to the agency. So a couple closing remarks. One is there's a lot to think about with medical device developments, especially with neurological physical medicine devices. Getting a reasonable assurance of safety and effectiveness, obtaining that data set. I've tried to provide some resources here to help folks navigate the landscape. We have a neurological devices webpage that's available online. I think it's one of the first ones available that goes through a neurospecific walkthrough of what the requirements might be for any particular product. 2, pre-submissions, right? They're free. They're free. They're really free. So you should use them. I mean, it's like a free way to get feedback of the agency. I'm surprised we're not like overloaded with pre-submissions because you spend all this money through the NIH, through NSF, through DARPA, through IARPA, DOD. But if there's no end game to getting that product to the patients, it's like lost. So you really want to have some engagement with the FDA. And 3, I always say this, if you didn't remember anything in my talk, and you still don't know what FDA does, there's my email address. Contact me, and I'm sure I can get you to the right person that is interested in your device area to get your device to the market and to a patient that desperately needs something. Thank you.
If you want to use a new device to deliver a drug, so then you have to get approval for the device and the drug, how does that work?
Yeah, so that's a great question. So let's say you have a delivery system that delivers a drug to the brain for brain tumor, hypothetically. We would work with our Office of Combination Products, and they provide support in determining the primary jurisdiction of a product to which center it will be reviewed. So in those cases where the primary mechanism of action is a drug, primary oversight will be with the Center for Drugs Research and Evaluation, CDER. And we'll be consulted because it'll involve a technology. So we have a lot of experience on the questions, the infusion rate, the placement of the product in the body, the materials that the product are made of. So we'll work with CDER on those, in those submissions that have both a drug-device combination. There are many times that those assignments get to the devices world. Ultrasound, permeabilization of blood-brain barrier for different drugs. That requires a different set of questions. But our Office of Combination Products will help us determine who's the primary lead based upon the mechanism of action. But behind the scenes, I talk to my colleagues in Division of Neuro a lot. We try to harmonize our policies and approaches to products so that it makes sense. We're going to have a little bit slightly different questions and needs, but there should be some harmony in both the drug and device and biologics world.
Thank you. Any other questions for Dr. Pena? I have a quick question. These days of use of the cloud for transmitting medical information and hacking and so forth, those devices that do connect to the internet, does your office get involved in safety in terms of the potential for hacking?
Yeah, that's another, that's a 21st century question. We have, I think, was recently cybersecurity guidance for our products, especially for like the DBS, deep brain stimulator applications that do these remote sensing and connecting with their products. There's guidance that's available for cybersecurity. We have software engineers. We have, excuse me, we have a number of different scientists at the agency. So when we get a new system, let's say a new deep brain stimulation system that comes with an IPG, the leads, electrodes, we'll pull together a team that can look at both the clinical outcomes, the non-clinical data, but also the software, the cybersecurity, the EMC, the other components that help to ensure the safety of that product for its use.
Thank you. I want to thank Dr. Pena. This is one of the few meetings where clinicians and industry have a chance to hear from and interact with folks from the FDA, and we're very appreciative of you being part of this course now for the third year in a row, so thanks. Our second speaker is Dr. Amir Tamizh. He is the director of the Division for Translational Research at NINDS. Those of you who were with us these past couple days, assuming you've identified your unmet medical need, put your team together, now it's time to get funded. And Amir is going to tell us this morning about funding opportunities through NINDS, which really run the full gamut from throughout the entire healthcare innovation cycle, including later in the cycle for support for spin-out companies and so forth. But this is a very important talk for those of you who contemplate applying to NINDS for moving your projects forward. Amir?
Thanks, everyone. Thank you for introduction, and good morning, everyone. I'll make sure this is worthwhile waking up for. So I do get reminded how amazing Carlos's team is, by the way. So we work with them very closely, as you may imagine, and Carlos and his team it's just been tremendous colleagues. They are very open, and the comment about pre-submission is something that we really encourage all of our applicants to take advantage of, because that's really the one time where you can go in and ask some really, really fundamental questions and get in front of Carlos and somebody from his team and get answers. So you are very well equipped for your actual IDE or whatever the next application may be. So I do encourage you to contact him. And I know for a fact that all of our applicants and all of our grantees that do work with them, they're very satisfied with the speed at which they get back to you and the professional nature that they treat everyone. So I'll tell you a little bit about NIH, and I try to parachute you into our organization from very high level. So the National Institutes of Health, we have 27 institutes and centers, and every one of them is distinct and has a very different mission. They offer funding opportunities that are very unique to the mission that they have. They have different budgets, different types of grants. There are various mechanisms, grant mechanisms, through which they use these fundings. They have contracts, they have grants, and they have other transactional authorities that are very unique and are very helpful. And just to give you a perspective, at NIH, we have actually more money for neuroscience than any other disease. So together with Aging Institute and mental health, our budget is actually larger than cancer. So we do have significant funds available to really spur science. So the mission at NINDS is pretty basic. We really want to understand the fundamentals of the nervous system and the brain, and hopefully use this science to help reduce the burden of the disease. Within NINDS, 90% of all the funds that we have, which is just over $2 billion a year now, we use for grants. And for that, we have three divisions. Divisions for basic research, which is a division for neuroscience. We have a division for translational research, and we have a division for clinical research. I believe that we have representatives from all these divisions here throughout ASENT, throughout the week, and I really encourage you to ask them questions and find out more about what we do, because at one point or another, you'll be interested in a grant mechanism as academics, or as a startup, you'll be interested in the SBIR program, which actually is a set-aside fund, which takes up about 3.6% of our entire budget. So if you can imagine, we have $70 million to spend on startups every year. And we issue approximately 100 grants a year to brand new startups. So there is significant opportunity for people who are interested in spinning off companies to really get involved and take advantage of some of the fundings that we have available. So what do we do within our division? So we're really interested in helping investigators advance their science to improve health and quality of life for patients with neurological disorders and stroke. And there are three fundamental strategies. One, we want to make sure that we have the appropriate programs, grants, contracts, screening capabilities, educational mechanisms, and various expertise through our cadre of consultants that we have, so that we can really help investigators that come to us. We emphasize going beyond publications. Publications are great, and it allows you to have tenure and do various other things, but for us, we really are interested in product development. And so we are really interested in see how a publication can actually turn into a product at some point. So that's where we get really involved. So if you're interested in conceptualizing what a product development cycle looks like, we are a step past a publication being published in a very high impact or even low impact journal that shows a promise for something to go beyond a graph. And we're here with the understanding that we don't really manufacture and commercialize drugs or devices, and we really are playing a role in the product development ecosystem, and we emphasize de-risking projects so that groups can partner up or license to organizations, industries that actually have all the resources for getting in front of FDA, getting the approvals, getting the labels, and manufacture and sell the products. Okay, so I think I've given some perspective of where we are. We are actually across the street on Executive Boulevard. We occupy the second and the third floor. And our division has many, many resources that I encourage you to look up. There are development programs, many of which are catered towards manufacturing, proof of principle studies, regulatory practices, and early clinical trials. Those are depicted on top. And because of modalities, we're splitting up our efforts for small molecules for Blueprint Neurotherapeutics Network. We have large molecules, including gene therapies and cell therapies through Create Bio. We have a very vibrant device program now, which we established about 2 years ago. And all of these modalities are actually covered within the SBIR program. And there are a number of feasibility programs that we offer. And the latest addition in that cadre of programs are the biomarker efforts that we established about 2 years ago to not only discover biomarkers, but also validate biomarkers. So that can actually do clinical trials to really fully validate prospective biomarker development. And we just launched a preclinical screening platform for pain to help spur the community to really get into this field and test their interesting compounds or devices in this program. This is what the portfolio looked like as of 2018. So as I mentioned, we annually offer about 100 grants per year, majority of which are really around small molecules. The device program has been growing very rapidly, and we are now really into model development, assay development, and things beyond what you see in blue and red and green. Here's a typical example of where we get involved. So here's a story for Susan Slogenhaupt, who discovered the gene that's mutated from familial dysautonomia in around 2000. And she took advantage of our regular mechanisms for R01 and R21. These are the 2-year and 5-year, 4-year grants that you typically apply for to identify a compound, kinetin, working with our colleagues at NCATS, and also developing a mouse model. So where we got involved is in blue, where we advance kinetin and its structural activity to really robustly identify a molecule that is actually a drug-like. It's orally bioavailable, and went further to characterize it and do exploratory tox studies for it to be really ready for clinical trials, at which point in 2015, PTC Therapeutics licensed the technology from Harvard. So we think ourselves as the bridging mechanisms to really go between the discovery efforts in R01 field to the therapy development, which is done in industry. Similarly, for gene therapies and oligos, here's a example for SMA out of University of Ohio State. So the engineering was done through an R01 mechanism. However, the delivery of the SMA gene and further structure work that was done and IND enabling was funded by us. This is a product that's been quite successful and has garnered a lot of interest by industry, which led to a partnership with Novartis. And the product has been coming out with a number of really interesting results out of clinical trials. So what are the key attributes for our programs? So we emphasize deliverables. We work with the investigators to understand where the endpoints are for these grants. We set up milestones and work through them stepwise to make sure that we stay on track. Many times investigators really overestimate what they need. So we are in position to provide supplements, to provide no-cost extensions, and also connect investigators with other programs that they can benefit from. These are very focused projects, so there will be very little overlap if, in fact, a PI decides that they want to do something complementary and they can actually get another grant. So we do have many of our investigators that have multiple grants. And as I mentioned earlier, we are providing many resources beyond the traditional grant mechanisms, contracts, resources that are completely in kind. So if you have a grant with us, you can actually, now you're in the system, you can benefit from many of the activities that we have. We offer training and workshops. So one of the training programs that is ongoing right now is actually downstairs. We provide a 4-day training for all the people that are interested into getting into product development, particularly for Blueprint, which is focused on small molecules and biologics. And we have many resources on our website describing target product profile practices, commercialization expertise for how to come into the SBR program, and various other things. So I'd like to focus on a couple of these programs and give you a little bit more in-depth knowledge about how they operate. So to start, I'll start with the Blueprint Neurotherapeutics Network, and this is really the program that got me into NIH in 2012. So here, the vision is actually quite extraordinary because we're really thinking about a world through which an investigator can have all the resources they need to advance small molecules. And what it entails is to not only provide them a grant support, but all the other resources they need and build a team around them so they can actually move their product forward. We can provide this resource anywhere from the left-hand side on the exploratory discovery side all the way to early clinical trials. And there's a preparatory phase involved where we just get to build a team. So we work with you and try to understand what are the things that you want to do and what are the things you need and you don't have, and try to build that around you. So a typical team consists of about 15 people. That includes the PIs, the lab, any core facilities they may have at the university, and has a group of consultants that we bring in, and has a number of contractors that we bring in that cover what's on the out rim of this circle. So we provide the medicinal chemistry expertise, we can manage all the data, we can do all the PK talks, we can actually do clinical trials. Currently we have 2 of them. We can do formulation and manufacture, and at the same time provide the grant funding directly to the investigator. So this is really unique, and that really got me excited about this, because this is something that I was really striving to do in private sector, and I thought this is really an awesome model, and it's been quite successful. We've had a number of successes and a number of products that has actually moved into clinical trials through this. Here's an example of how we can get involved. So typically academics don't have resources for formulation and manufacture. Here's a perfect example of a compound that was being developed by Paul Kenny at Mount Sinai, and on the left you see how in his in vivo studies he was not really seeing any differentiation between any other doses compared to vehicle, all the way to 100 mg. And after working with him for a few months, we realized that what was happening was that as he was increasing the dose, the drug was just not getting a solution. And as part of this, when you're injecting this suspension into an animal, you just don't see any effect. So working with our contractor, we actually came up with a formulation that's depicted in the lower panel on the left-hand side, which uses a significant experience by a formulation group that really understands how to deliver things in vehicles. And as you can see, that result changed significantly once we actually had the right formulation. So it's not about just the right molecule, it's about how you actually make it a drug. So we have these resources, we provide them in kind, and once you actually are within our system, you can have free access to them. Likewise, as I mentioned before, we can do more than just typical manufacturer. Here is a project where we realized target engagement was very critical, and understanding of pharmacodynamic measures was going to be really important for us to better understand how we can manage expectation in clinical trials. So we offered this investigator a resource through another institute to actually do PET imaging and study target occupancy and better predict efficacy for this compound. This compound is actually in phase 2 clinical trials now in a few indications. It's been quite successful. Having done this has been really tremendous amount of work, but at the same time it's been really rewarding, because now we can actually be much better informed in the dosage and the duration of this drug once it's actually been administered. Likewise, here's another example just to give you a quick perspective about the timelines and how fast we can move. This project by Mark Gurney, looking for PDE4D inhibitors, came into Blueprint Neurotherapeutics Network in 2012, and by 2015 we had completed the IND, and we actually finished up a phase 1 trial. And the capsules you see on the right-hand side are actually of the picture of the capsules that we dispensed early in the morning at the clinical site. So we understand, many of us have worked in an industry, we know the speed at which we need to move, we know the patent timeline is in play, and we really understand how patients are truly waiting, and we can't really have a system that's just not efficient. So we think based on the resources that we provide, we're quite efficient, we can move quite fast. I have a movie here that I'll share with you. It only takes 2 minutes, so I'll let Susan Slogenhaupt, whose story I told earlier, to talk about live about how the resources came through. And I hope the voice comes through here. Is there a chance you can increase the volume? If you can maybe put the mic right next to the, maybe you can just follow it by just reading the text. So the take-home message from this is really to appreciate the fact that Sue is a biologist, pharmacologist, has been studying familial dysautonomia for years, and within her circle, she didn't have the resources to really push this compound, kinetin, through. So by building a team around her, providing the expertise with medicinal chemistry, regulatory, and scale up and manufacture, we were able to really bring the drug forward towards clinical trials, which is something quite unique. It goes well beyond what you can do with the regular grant mechanism. Similarly, we have a program, Create Bio, that's led by Chris Boshoff for biologics. And that really covers peptides, proteins, oligos, genes, cells. This program is quite different in respect that we don't provide the medicinal chemistry, obviously, and we really don't have the appropriate contract mechanism to do all of these in one shop. So what we do here, we provide the funding for the investigators to actually identify, and we work with them to identify the appropriate CRO or a collaborator who can actually do this. It really covers target validation all the way to early clinical trials. With biologics, we understand that the track is a little different, and we provide two different entire grants, one for optimization track that really looks at various modifications you need to do for the biologic, and the development track, which is truly an IND-enabling track that ends with a clinical trial. There you have to bring your product to us. We don't really do any optimization. Together, this covers anywhere from $6 to $10 million worth funding that you can get access to. But very much like Blueprint, we provide consultancy and regulatory resources to sort of trek through that environment, work with teams like Carlos, and figure out exactly what we need to do. Here's a quick snapshot of what the program looks like. So we do a significant amount of investment in gene therapy. We have oligos. We in fact have cell therapies, and the disease areas are depicted on the right-hand side. Currently, the program has about 15 grants and 15 projects, and every 4 months, we're looking for more projects. So if you think you have the next biologic for a neurological disorder or stroke, come talk to us. What I just mentioned for small molecules and biologics is all covered with an SBIR program also. So if you're a small business, in either stage, the stage where you're doing tech transfer, working with an academic PI to really start a company, which is the STTR phase, or you have established your small business and you want to do any of the activities that I mentioned earlier, you can tap into a set-aside fund, which is very different for us. And so you can have the Omnibus SBIR program to take advantage of, or you can come into a companion program. For example, we have a companion grant mechanism for Blueprint that's using the SBIR fund. So it's very similar, very dynamic, and we work with you to try to figure out exactly where or what program announcement you can actually benefit from. So let's shift gears and go back to the previous stage, before you have your compounds or before you have your projects. So Becky Roof joined us about 2 years ago, and she's leading a program called Ignite. And the strategy here is threefold, really. It's to sort of work backwards from blueprints and creates to figure out what a successful project look like. And we realized that for a really good project to come into those programs, we needed to have a hit molecule or a lead molecule, however you want to describe it, but something that we can work on. We need to have a reasonable data package for proof of concept in vivo. And ideally, you want to have something beyond just in vivo efficacy studies, perhaps a pharmacodynamic measure, additional PK study, to really understand where the initial hit compounds are. So there, appropriately, we have designed 3 program announcements that really cover all those 3 areas. These are 3-year grants. They use a mechanism that's quite unique. It's an R61, R33. So there's 2 phases to this. One is a preparatory phase. It could be anywhere between 1 year to 2 year, and an execution phase, which is complementing the rest of the 3-year cycle. You do need to provide us with what you will do as part of milestones before you go from preparatory to execution. For example, if you're proposing to do an assay development and a screening effort, at the end of your first phase, you want to make sure that you at least have your assay up and running before you can do the screening. So that's really what it is. It's just trying to make sure that the appropriate strategy is benchmarked, and we will work with you to figure out what that is. Here's a quick snapshot. We currently have 33 grants in this program. As new as it is, it's actually been stunning how fast it's growing. It covers many modalities on the left-hand side and many indications that we have on the right-hand side. Again, we've been awarding anywhere from 4 to 7 grants every cycle on this, and we're hoping that this actually can fill the gap between the R1 space and our later development phases. Here are some examples for the type of work that we're doing. Just want to highlight the fact that Jim McNamara has been developing TREC1B activators for epilepsy. He just graduated from this program. He successfully not only developed the assay, he identified hit compounds, and he was just awarded a Blueprint grant. So he's hitting the ground running with significant resources in medicinal chemistry and so on and so forth. So this is exactly what we want to see. We want to see people come in here if they don't have all the T's crossed and all the I's dotted, but once they graduate from here, they can move on. So we're committed to actually developing things towards clinical trial, and we feel that we have a lot of the resources available now. So when I took the helms at the office, I had the idea of really doubling down on biomarkers, and we were fortunate enough to have Marianne Pellymounter on the team already, who has had a lot of experience in industry and has really led the effort. But the idea here is to really facilitate therapy development by combining it with the appropriate biomarkers that go beyond the endpoints to really facilitate clinical trials. Here's recent publications to really justify and really show how important biomarkers are. And as you can see in the graph, the way it's depicted, every time you have a companion biomarker in any of those studies, whether it's phase 1 to phase 2, 2 to 3, and beyond, you do get an advantage. You do have a significant improvement in probability of success. And we felt that this was an important thing to do. And it goes beyond just clinical trials. So here, we're looking all the way from basic research all the way to approval. And as you can see, even in basic research, there are many places where having a biomarker really helps advance and accelerate therapy development, whether it's for identifying the mechanism of action of a study, of a drug, target engagement, and various other activities that you can do in clinical trials. So we feel very strongly that this is really the way of making best use of our funding. So accordingly, we have 2 grants, one for analytical methods development and one for prospective clinical trial studies. The reason why you see 2 different PAR numbers there is because we are doing this as part of the regular grants and also as SPARs. And just very recently, we took advantage of the HEAL initiative, which is tackling the opioid crisis, and we are doing very similar programs for pain. Now I want to shift gear away from the type of grant mechanisms we have to just in-kind resources that we have. So John Keene leads the epilepsy therapy screening program. This is a program that is in existence for 30 years and has led to number of identification of new therapies and actually approval of therapies. And it's quite unique in that you don't need a grant to come into ETSB. You can actually contact John, and based on the type of information you can provide to him, you can actually come in and test your compounds in our animal models and in vitro models for epilepsy. Over the last couple of years, we've shifted gears a little bit, and based on the council working group recommendations, we've now moved away from some of the early models that we had adopted starting in 1970s to much more of disease models. So now we have a cadre of acute models, pharmacoresistant models, and disease-modifying models. The program is done through a contract in the University of Utah. And for all of you guys who may be interested to learn more about this, you'll have one more chance on October 9 to actually visit with us in Utah during our open house to learn more about how we run the studies, who runs the studies, and get much more familiar with the program. So not only we provide the in vivo pharmacology work, but we also have a database that's been quite popular, and Panache provides the data that's been generated at Utah, but also provides all the protocols, all the standardization, and all the best practices that one may want to tap into when you start a lab or when you think about moving into epilepsy. So all that is publicly available, and it actually includes over 200 compounds now with all the data associated with it. And very much like ETSB, we are actually launching a brand new program for accelerating therapies for pain. So we think that we can actually duplicate HTSP and the successes we have benefited from and really generate high quality data for pain models. Here is a tier mechanism that we are thinking about starting, and actually the website just got launched yesterday, and we have a RFP for a large contract that we would like to award to actually do this work. So beyond that, I want to take advantage of the few minutes that I have to talk about some of the other programs that we have as part of the pain initiative, the opioid initiative. So again, if you look closely, we have this as part of the SBR program, set-aside fund, and the regular grant. So we have 4 programs that I'd like to highlight here, all of which you can actually find within our website. So the first is a optimization effort for non-addictive therapies. This covers small molecules and biologics. And similar to Blueprint, we provide all the resources through contracts, so we can actually do many things beyond just the grant funding that we provide to the investigator. Likewise, we can do this for devices. So we actually have a MOU with Carlos and his lab. So whenever a project comes into our translational device project, we discuss them with Carlos and we provide that resource to all of our investigators. It's been actually quite helpful to really accelerate the timelines and get real live feedback from Carlos and his team to what the next steps may be as part of advancing the device projects. We do provide the resource for biomarkers and retrospective studies for analyzing some data samples that you may have from a clinical trial, and prospective studies for studies that can actually take you into phase 2 clinical trials as part of a drug study or all within itself. And that covers many modalities, if not all modalities, including imaging and various other modalities that you can have. And that actually has garnered a lot of interest. We are hoping to go well beyond the mission of NINDS in this case. So as some of you may know, the HEAL initiative, Help Addiction Long Term, has been a cross-NIH effort. And this program has 16 partners, including the Cancer Institute and various other institutes that are really squarely within the pain space. And we can actually manage these grants on behalf of the other institutes. And we're really excited about this. We just had our first receipt date, and we've gotten very, very interesting projects to move forward to. This is a 5-year congressionally mandated funding that we have just for pain and opioid use, and we hope to really change the landscape for how we do clinical trials. And biomarkers are going to be very important. Okay, so I've said enough about all the other things that we do, but how do you actually come into our program? So here are a couple of the bullets that I'd like to leave you with. So it all starts with just learning about some of the programs that we have. So you can learn about them just by talking to us, sending us an email, or looking at our website and get the information that you need to proceed. I really encourage you to talk to program directors. Many of us are walking around here today, and just stop us and just ask some of the questions you may have about the programs. Beyond this, you can send us an email, you can call us. I keep telling everyone that our job is actually to talk to our investigators. So I make sure that I am available, and all of our program directors have been actually quite progressive about this. We typically ask that you come to us with a one-page summary of what it is you try to do. We see hundreds of these, so we should be able to very quickly look at them, set up some time with you, and go through the proposal. If the application that you're trying to do is above $500,000, at NINDS you actually need a initial permission to come in. So at that point, you really do need to talk to us to make sure that you have the appropriate approval to move forward. That typically takes anywhere from 60 to 90 days. Don't worry if you're not funded the first time. So the funding success rate right now at NINDS is somewhere about 20%. And as an FYI, new investigators take a significant advantage in our system and go well beyond the pay line. So the pay line for NINDS for this year has been about 16 percentile, but we pay out all the grants for new investigators up to 25 percentile. That's a big increase, and I encourage you who are just getting into the program to really take advantage of that. If you don't get funded, it's quite all right, actually. So it turns out that many investigators only apply one time, and they never come back to us for various reasons. But the success rate dramatically increases in your favor if you come back. And the way you can do that is by carefully reviewing the summary statement, sending up another call with the program director to go through the summary statement in detail. And the next application that you come in with provides you this one page to do the rebuttal and to identify all the things you would do differently and how your new application is quote-unquote an improved application who's addressed all the weaknesses from the previous application. So I've seen the success rate go up dramatically in what we call the resubmissions or A1s. And I encourage you to think about that as a way of long-term planning as opposed to a short-term plan where you say, well, NIH didn't want to fund my project, I'm just going to take my business elsewhere. That's, to some extent, is what we get. Lots of investigators that are just getting started don't come back. With that in mind, so here's a process. So initial application, you want to talk to us, especially if you're a small business. You want to understand how we do some of our programs. Once you apply, the application goes directly into the scientific review. From then, the summary statements are generated. Program directors typically will have a chance to talk to you about your application and be best informed when they actually take a pay plan, which hopefully includes your application, to our advisory council. The advisory council is the second body of review that actually approves our pay plan. And once that's happened, within a few days, you typically get an email from one of the program directors saying, your application is ready for funding, and within the next few months, we'll work with you to find out what the next steps are. So these are the timelines, just very quickly. About a month or two for a review, the initial review, for about a 3-month cycle to get to council. So we have council 3 times a year. So once every quarter, actually a little more than that, we have an opportunity to take a pay plan to our council, get the approvals, and pay out the grants that we want. And within 2 to 4 months, the funding is available to you. So I've said a lot. I'll stop at this. Happy to take questions. Again, just like Carlos, I really encourage you, if you have questions, if you want to explore what we do a little bit more, just send us an email, and we'll make sure that we'll get back to you, or more likely actually direct you to the right person to talk to. So if you send me an email about Blueprint, I probably won't be the right person to talk to. So what I do is I'll connect you with Chuck Sywin, who can then get back to you and set up some time to talk to you about Blueprint. And that way you can make much more informed decision. We tweet, we constantly update our website, and as an example, the PSPP program was just launched yesterday, and I encourage you to look at it. It's a wonderful new program that we're really excited about, and we're hoping to play a big role in the landscape for pain. Thank you so much.
You didn't say anything about sort of next level, NeuroNext, and what role that plays.
Yeah, so anything beyond phase 1 for SAD and MAD, which we cover, there are really 4 different ways now to really think about how to do clinical trials. One is through a regular grant. The other one is to come in through some of our center programs that we have set up, one for stroke, one for any neurological disorder, which is called NeuroNext, and we are hoping to launch a new program called EpicNet for pain studies. So talking to the program directors would be helpful, so then they could direct you to where you can go to get additional resources. So there, you do get a grant again to be part of the study, and we'll provide you the patients and the capabilities to actually execute clinical trials, which is quite significant. And currently, we have anywhere from 5 to 10 clinical trials within each one of these programs.
Great. Any other
Thank you.
questions? Thank you so much for coming, and thanks everyone for coming so early this morning. So ends our course on translation. Thank you.